So I'm going to go ahead and work a little bit on host defenses too. Let me ask, are there any questions about the lab? Anyone have any questions about the lab? So in the lab, remember, you should finish, actually you guys have already finished lab 9. No, you haven't. You should finish lab 9 this week and be working on your unknown project work and your infectious disease project work. The infectious disease project is due in just a few days, 7/14. The unknown project is due to 7/21. I sent out yesterday a number of tests, so some of you got tests yesterday. I hope I will be able to look at that sometime tonight, but it'll be late tonight because I'm teaching another class, let's see, starting at 5. So, but I hope to get to some tests that were requested and should be available today. Remember, when you request a test, you can look up how long that test must run, and that test, while it's running, it's incubating, and you're not going to get your results until the test is done incubating. So some of you made requests like 2 days ago, and those requests, if they take 2 days to incubate, you should get it today if I have time to get to it tonight. And if not, I'll get to it tomorrow. Those of you who requested 2 days ago, if the test requires 5 days, you're not going to get it until after 5 days. Any questions about any of that? Anyways, be working on your lab. Try and finish up your infectious disease project and then work on your unknown projects. And like I said, I want to work a little bit on host defenses too. And so we will get out of class early today. Now where do I have that? Load that up. So the learning goals for host defenses 2 and a slight rough outline. Be able to compare and contrast the humoral and the cell-mediated immunity. These are 2 arms of the immune system, and so you have to know about the humoral and the cell-mediated immunity. Be able to list the results of what happens when an antibody binds to something, and what are the results that can happen when the antibody binds. Be able to describe the various structures and the various kinds of antibody molecules and their functions, meaning when we make antibodies, we make 5 different antibodies, and they have a different structure and a different type of antibody, and the different types have different functions. Be able to know how immunity can be artificially acquired, naturally acquired, and be able to differentiate between when the immune response is active immune response or a passive immune response. So immunity can be artificially acquired or naturally acquired, and be able to differentiate between when the response is active or when it's passive. Be able to describe the different types of T cells and their functions, and be able to differentiate between a T-dependent antigen and a T-independent antigen. Any questions about any of that? Okay, the last time we talked about the non-specific types of resistance of the body to an infection. We mentioned that there are different mechanical and chemical factors that serve as the first line of defense, and that involve the intact skin, the intact mucous membranes, and then the secretions, actually both by the skin and the mucous membranes. Remember that the skin does secrete sweat, and there can be the enzyme lysozyme in it, as well as sodium chloride. And the skin can also secrete sebum, which that's a very thin barrier to infection. It's almost so thin that it's not much of a barrier, but it can be metabolized by the normal biota of the skin to help defend the skin. And then you know about the mucous membrane secretions. Obviously there's mucus, which is a barrier to infection. If an infective agent gets on the mucus, it has to get through the mucus, which if you got lots of them, it's not a problem. But if it's one, it could be a problem. And then there are some enzymes like lysozyme also in many of the mucous membranes. Okay, any questions about any of that? Today we're going to move on from the non-specific defenses and talk about a specific host immune defense, or the third line of defense. When we talk about a specific host immune defense or the third line, we're talking about the same thing. And this is the host immune response to an invading pathogen. Okay? The host immune response depends on two specialized lymphocytes, and we'll talk about them. They are the B cells and the T cells. And you can also call those B lymphocytes or T lymphocytes. Any questions about any of that? When we say it's a specific defense, we mean that it is acquired, meaning the body learns to respond to that agent. Like, for example, unless you've already been exposed to COVID-19, you probably have no specific defense against COVID-19, which is why it's so, one of the reasons why it's so dangerous, and that's the reason why so many people can get it, is that humans have never seen it before, and so until your immune system learns to respond to it, it doesn't. And we'll talk a little bit more about that. But that's what we mean by a specific defense, and that is your immune system learns to act against it. And what it's doing is it's mounting attack against specific molecules of the pathogen, in this case COVID-19. And what specific molecules we're talking about are generally the external molecules on the pathogen, meaning the host doesn't see molecules, or I should say generally the host doesn't see specific molecules inside the pathogen, like inside the cytoplasm, or if it's a eukaryotic pathogen, the host doesn't see the molecules inside the nucleus, but it does see the molecules on the surface of the pathogen. And those are the molecules the host will respond to. And so when we're exposed to a new virus, we have no specific resistance to it. You can mount a non-specific defense, like your white blood cells will respond to a viral infection, and they'll try and engulf them. The trouble with viruses is, of course, they infect your cells, and then the white blood cells can't act. And we'll talk about how different host cells can act against a virus once it's infected its cell, but we'll talk about that later in the lesson. And you can't respond with a specific defense until your immune system is learns to respond to it. And what happens is the immune system can mount a specific immune response against the antigens of a pathogen. Antigens are almost always a cell surface molecule that the host responds to. And so the antigen on this cell, this looks like a eukaryotic cell, is in the cell membrane. And here's the antigen here spanning the membrane. And then the host specifically reacts to certain parts of that antigen, and we call those antigenic determinants or epitopes. And an easy way to think about that is when the host makes antibodies, different antibodies could react to this antigen, and one antibody could bind to the cone-shaped molecule on the antigen. Others could bind to the semilunar molecule on the antigen, and then others could bind to, what is that, a pentazoid? Okay? And so you have 3 different antibodies that the host is making, and they're all reacting to the same antigen, but they're reacting to different parts of it. Do you see that? So most antigens are a protein or a large polysaccharide molecule. They can be other things, but most antigens are large proteins or large, actually just proteins, or because most proteins are large anyways, are large polysaccharide molecules. And that is what the host will respond to. Remember, when we're talking about this antigen, the host will react specifically to one part on that antigen. So they're not reacting to the entire molecule. They're only reacting to a part of that molecule. And different antibodies, for example, could react to different parts of the same antigen. Of course, they can also react to different parts of different antigens. And we'll talk a little bit about that. Lipids and nucleic acids are not very antigenic unless they're combined with a protein or a polysaccharide to make them more antigenic. So lipids and nucleic acids can be an antigen, but they're not very antigenic. And I'll talk about what I mean by more antigenic. It means that the host evokes a stronger immune response against an antigen that is more antigenic than it will against an antigen which is less antigenic. Did I confuse anyone yet? And so the more antigenic molecules tend to be proteins and large polysaccharides. That means most antigens have a molecular weight of 10,000 or greater. And that probably doesn't mean a whole lot to you, so let me give you a comparison. Water, which is H2O, sorry, is a molecular weight of 18. So water is a small molecule. And then penicillin, this is a molecule we're talking about penicillin G, a molecule we talked about a lot earlier, which we consider a large molecule, has an antigenic weight of only 302. Okay? So what we consider a large molecule is not large by what the host immune system considers your average antigen. So it would be a very large protein, which most proteins are large, by the way, or a very large polysaccharide. Small molecules are not very antigenic, and to become antigens, the small molecules have to attach to a bigger carrier molecule, such as a molecule in the blood. And this is exactly what happens when people become allergic to penicillin. The penicillin binds to a larger blood protein, and then the host immune response, meaning the allergy to penicillin, happens because the penicillin is bound to a larger protein in the blood. Is that clear? So small molecules can become antigens by binding to a larger host molecule. And then the host reacts to both the small molecule and then the larger molecule. And that's how people become allergic to penicillin. We're not really going to talk much about that, but I'm just telling you that's how penicillin, people can become allergic to penicillin when it's such a small molecule. Molecules of invading microbes can act as antigens, and these molecules tend to be molecules on the external portions of the pathogen. So that could be in the capsule, if they don't have a capsule, in the cell wall. Flagella, if they have a flagella, remember that extends out of the cell wall. You can also react to toxins that are secreted by the pathogen. We haven't really talked much about toxins yet. I don't know if we ever will. Maybe I should try and do that in this lesson. But some toxins are made by pathogens and then they're secreted. We call those exotoxins because they're secreted. And the immune system can react to those toxins and inactivate them. And of course, if you have a naked virus, the antigens of the naked virus will be the protein coat of the virus. For a lipid surrounded virus, meaning an envelope virus, it's usually the external parts of the envelope. And for most viruses, that will be the spikes. And for COVID-19, it's the spikes of COVID-19 which our host immune system will respond to and recognize as foreign. And so they will make antibodies to the spikes of COVID-19. So in the figure, we have a cell membrane, and this antigen is a molecule in the cell membrane, and it's not phospholipid. It's probably some large protein in the membrane. And the host is recognizing this as an antigen. But, like I said, it's specifically reacting to one part of this antigen, meaning the antigen is the entire molecule, but the host is specifically reacting to one part of that molecule. And that's what an antigen is. Any question? So what the host is specifically reacting to is called an antigenic determinant or an epitope, the specific part that the host is reacting to on this antigen. And that would be what the antibody would bind to, this epitope, assuming that that antibody binds to that epitope. And the immune response is started by a lymphocyte. Any questions about any of that? Everyone's good? All right, well, let me just remind you that we looked at the lymphocytes. Those are the white blood cells, and the lymphocyte is the second most common white blood cell. When you're looking at it under the microscope, you can't tell, is that a T lymphocyte or a B lymphocyte? But it's the second most common white blood cell. It's one of the kinds of white blood cells. It's mostly a nucleus. It's an agranulocyte, meaning it doesn't have granules in the cytoplasm. we say that there's two types of lymphocytes, and I should state that there are really only two main types of lymphocytes. There is a third type that we are going to talk about very briefly. They're called NK cells, and they are involved, NK cells, they are involved in the immune response, but they're non-specifically kill extracellularly, as do eosinophils, meaning an NK cell comes up to another cell, which is either a pathogen or one of the host cells that's infected, like with a virus, or it might be a host cell that's become cancerous, and then the NK cell extracellularly secretes substances on that cell to kill it. And we'll talk briefly about that later on. But most of the lymphocytes we're going to talk about are the B and T cells, and they start their life in an adult from the bone marrow, and I think we talked about that. That actually all the white blood cells start in an adult from the stem cell in the bone marrow cell. Okay? Any questions about any of that? If not, this is a good place to start, and I'll say next time I will start talking about T cells. Okay? If there's no questions, I'm going to end it now, and I'll see you next Monday. Okay?